point of care diagnostic to see that they are real-time, you have real-time connectivity, ease of specimen collection, they are affordable, sensitive, specific, user-friendly, rapid, and robust, equipment-free, and deliverable to end users. If you satisfy these criteria, then you can classically be able to have, say you have this point of care diagnostic that can be used in very diverse settings, be it, we've seen some of these tests even being used at homes, okay? The classic example really is the pregnancy detection test, the PDT, and we know that women have been using this test at the comfort of their, bedrooms and bathrooms. Okay, so having an antigen lateral flow assay is great because it's cheap. You can get a quick turnaround time, and the problem is it has a high limit of detection, which means you may not really be able to detect asymptomatic infections. This was one of the biggest problems with the COVID-19 antigen tests. If somebody, and we knew that many patients, many people who had COVID were not, they were carrying very low viral loads. They were not even aware that they may be infected. You remember this scenario where even children were accused, and the young people were accused of spreading the virus to older people, but the young people were fit, they were okay, they had no symptoms at all. So this problem of high limit of detection then poses a problem there because transmission will continue. About ID Now and SafeAid, the expert tried getting these molecular-based tests, which is just an iteration of their other tests like the TB test, and they are very reliable, sensitive, and specific, with less than 1 hour turnaround time, but requires expensive instrumentation. So there was this report on, where people have previously used this CRISPR-Cas-based detection methods as a signaling method. And here, basically, we have these two strategies, the Sherlock and the detector. And basically, if you have this Cas enzyme with the guide RNA, if the guide RNA identifies target sequence in the sample or DNA, you end up with activation of the enzyme, and the enzyme will now start cutting, collateral cleavage or collateral cutting of other double-stranded DNA sequences, which are usually put in the reaction, and these will generate, a signal that you can read using a lateral flow assay. So we took this assay and combined with our previously developed iFAST device. iFAST means immiscible filtration assisted by surface tension. So we've, in our lab, working with our collaborators, we've developed this chip here where you just put your sample in this chamber here. The chip is this size, it's quite small, so it's portable and is made of plastic, so you can imagine the cost is not much. And you put your samples here, and then you can pull using a magnet, this is a magnet, through different wash chambers. And by so doing, so in your sample chamber here, you can, if you want DNA or nucleic acids, you can first of all burst open the cell membrane and the nuclear membrane, and then expose your DNA, and then bind it with magnetic silica or magnesil, and then move it to with washing to the last chamber, where now you can mount your amplification techniques, be it lamp or whichever. So just to show you, so here you have your analyte of interest. So you've burst it open, and you use magnesil or other reagent which can bind your analyte, and then using the magnet, now you move it all the way to the last chamber, and it is in the last chamber now you will now proceed to do your reactions as I will demonstrate. So we've used this strategy for detection of E. coli, this is published work. Group B streptococcus, this is also published. And we've done it for SARS-CoV-2 using normal color change reaction on the last chamber here. So this is just the same chip, but an iteration of it. So you do, you get your anal specimen here, or other sample, and then you move it through different wash chambers. Then you do lamp on the last one. And with the classical lamp, where people use turbidity or color change, you can use the same method to detect. And this work is published. But we've also done the same for Neisseria gonorrhea. And currently we're also exploring it for fungi. And so here, we leveraged on this to do iFast combined with CRISPR. I just mentioned about CRISPR detection. So we combined the iFast because one of the major handicaps with nucleic amplification techniques is that sample preparation is difficult. You only may be able to do it in mainstream labs where you have centrifuges and expensive equipments and thermocyclers and so on. So here, our chip is able to do these steps. You can do extraction, DNA extraction. You can do amplification on chip, okay? And you can do detection on chip. So that's a big benefit that can help you to achieve the reassured criteria. And then, so this just explains the science behind it. And you can then go ahead and use lateral flow assay on the last cartoon there to be able to visualize. Okay, we know we've been dealing with the lateral flow assays for malaria, for HIV, for pregnancy detection. So this is not really the learning curve is not there really for our healthcare personnel and even for lay people. So this is a strategy that combines all this to achieve that. Okay, so just more on the same, explaining how it works. That after, if you have a negative test, you just see the line on the control. But if you have a positive test, the enzyme, the Cas enzyme is activated and it does collateral cleavage. And then you have the quencher and the fluorescence dyes binding to the antibodies on the test line, and then you'll see a signal. And this is just these are comparison tests with the normal gel in the lab, just to show that to show the comparison between our test and the gold standard. So. And so this is just more data on that. Yeah, so, and different processes there, basically comparing for validity. And then multiplexing. So you remember there came a time when for us to really say we have this SARS-CoV-2, we need to show presence of, let's say, E gene, M gene, N gene, and so on. So our test, we thought, how could we achieve this? So we fabricated the test that now had three lines. Okay, so sample is one, and you do extraction here using chemical like guanidinium hydrochloride, and then you move. Okay, and then on the last chamber, your primers help you to differentiate whether you want to target E gene or N gene or whatever, M gene, okay? And even you can have a negative control as well, okay? And then another question that we had was, how can we, as I mentioned, we are developing this test for our settings, okay? So you want a situation where you can pre-store reagents on chip. So as you ship this chip to Meru, it already has the reagents there. So we lyophilized this and then did some experiments to show that you can lyophilize the enzymes and the other reagents on chip, and then just chuck them out of shelf, put your sample, hydrate, and then move to do the experiment. So this was really encouraging. So we showed that you could detect this on day 7. And subsequent work has shown that we could even proceed to around 42 days. So basically, further development of this test means we want to go into longer periods, as as long as a year. Okay, so basically we were able to integrate all essential steps that goes into a molecular test into one device, as I just explained, with a low limit of detection of approximately 100 copies per mil, which is very competitive compared to RDTs, for example. And we could detect engine and in 1 hour, but also with the multiplexing, we could detect even the E gene as well. And so we've been using just heating block in the lab, and heating blocks can be found in many labs. They are not expensive, okay, and also we lyophilized. So this is just another project that one of my master students just finished, and they just graduated last week. So we've also gone ahead to use hybridization chain reaction combined with CRISPR-Cas 12A detection as an enzyme-free detection for SARS-CoV-2. So this is work that is recently published. You can just check it in the journal Diagnostics. And also, because we want to reduce reagents, we want to reduce the cost. Enzymes are expensive, so if we can do something without enzymes, that's a plus. So hybridization chain reaction is a good candidate for that, and we've used it successfully to show that you can amplify this test and visualize it using Cas12a. The last project I'll talk about is the identification highly sensitive tools for malaria. Malaria is one of the major themes in our lab, and we know the classical molecular tests. They just look at a few repeating sequences, and then you amplify these and show on a gel, okay? But we have a new algorithm that is able to detect many sequences using the same primers, forward and reverse. You can detect a lot of regions inside the genomes. So for instance, if you look at PF, Plasmodium falciparum, the primers, the reverse and forward, same primers can detect up to 172, can amplify 172 genomic regions. Okay, this is really crazy if you think about the product you end up with at the end of the day. So basically you have, this is the situation. So these are the chromosomes in the PF genome, and with these primers, which are here shown as blue and red, you're able to amplify at many regions. And when you run this on the gel, you end up with, because the products, the amplicons are of different sizes, the classical IMRS gel has many fragments because they are of different sizes. Some are 100 or 150, 200, 300, and so on. Okay, and we know the classical 18S just shows you a distinct single band, as shown in that last gel image there. So you can see already with this, even without saying anything else, you see that with the IMRS, you are able to have a huge amplicon yield, increasing sensitivity. So, and remember we talked about asymptomatic infections, which are usually hard to detect because you have fewer pathogens or parasites in this case infecting the person. So you want a test that can really bust the few parasites or pathogens in the patient. So this just shows the promise of IMRS. And as you can see from the last probit analysis there, using IMRS, IMRS, which is isothermal, you can be able to, it competes against the 18S PCR. So you can see if you're having an isothermal test that is better or equal to the PCR, certainly you can see the deployability there. Okay, so this is just the same written in other words. Okay, and lastly, I know how many minutes do I have? Oh, super nice. I'm doing very well. Okay, so The last thing I wanted to talk about is, so last year, 2021, towards the end of 2021, we were fortunate to get a grant together with one of my collaborators from Stockholm. This grant is called Farmers Grant, and we founded this AFIA DX network. So the AFIA DX network basically aims to boost partnerships between African and European researchers working on development of point-of-care diagnostics. So we want to boost this collaboration, and one of my major motivations for coming here is to come and make connections and see how we can work together in building a partnership, strengthening this partnership, so that we work together. Sometimes it's really a shame that we work very well with people abroad, but not from home. So it's not great, isn't it? So we want to build to strengthen and have more partnerships locally. So in 2022, we had more than 10 webinar sessions with 24 speakers and around 200 unique participants. So we would invite people to come and just share their findings and ask questions and so on. And so we have a link. If you go to LinkedIn and you just say AFIA DX Network, you'll be able to see our page there, and you can go ahead and join. You can follow us so that in case we have any upcoming things, you can join. We've had these webinar topics, okay? Stories of success, overcoming challenges, dare to try, conquering hurdles, and so on. Basically, we have had people who are developing, from researchers doing asset development and prototyping, present their work. We've had industrialists, people who manufacture these tests, coming to share their work. We've had people who use them as clinicians, share their work, and so on. So it's really a nice platform for us to meet and discuss. So we learned some lessons from the AFIA DX network webinars in 2022. There's a big interest in POCD, research and development, okay? And therefore it means there is good impetus for collaboration. Multidisciplinary approaches are the best to tackle the myriad of challenges. As you all know, our commercialization is not so great, but our regulatory frameworks are not so clear. We have funding issues and so on. But if we come together, we can lessen these challenges. And working with end users is always great. It's always nice to know what do the clinicians want. Don't just develop a test in the lab alone, and then when it comes to the lab, the clinicians tell you we don't see value for it. So I want to stop at that, and I want to acknowledge all our funders and collaborators and our team members. And I would like to invite all of you to come to Mount Kenya University and visit our newly refurbished lab with the support from JICA. Thank you very much.
Thank you. Thank you very much, Professor Jesse Kitaka, for the good and technical presentation that you've shared with us. It's about new ways through which we can be able to detect and diagnose various diseases. And at this juncture, may I open it to the floor? So if there are any comments or questions, then a prof can be able to clarify to us. The floor is open. Yes.
Thank you very much, Prof, for a great presentation. So first of all, I would like to commend the type of research that you are doing. in a lot of forums we go, we see a lot of observational studies, especially in health, for those of us who are in the health space. So it's really nice to see where R&D is actually happening in a Kenyan university at a very high level. So I have two questions. First, on the iFast. Yes. I saw you have a number of chambers, and there is a magnet pulling the beads. Yes. So I'm wondering what happens in the intermediate chambers before it gets to the end, where it's being detected. So that's the first one. The second one is, you mentioned somewhere that enzymes can be quite expensive. I'm wondering if there is a way in which you think we could actually start to manufacture enzymes in our setups, because it's actually quite straightforward. Because many other assays actually require enzymes, even if it's NGS, the master mixes actually need enzymes. So do you think that it's something worthwhile for people in our setups to get into, for purposes of actually creating a critical mass of people who can manufacture enzymes? Thank you.
Super nice. Do I address those ones first or wait for more?
We can take 2 more questions or comments.
Thank you, Prof. My name is Dr. Amos Omamo. I'm the Director of Research.
Good.
I want to commend you for the fantastic presentation. Mine is just about the collaborations. I just want to get your feel. You said that you've done a lot of webinars, you've done a lot of sensitization, but what are the key challenges that you find? Because you say you're comfortable with the outsiders. But insiders, how do you build that critical mass? And where do you get the funding internally if you don't get it outside?
Excellent, excellent questions.
Thank you, Professor. My name is Odhiambo, the Vice Chancellor. Really want to appreciate you for coming to this university.
Thank you, Prof.
And it's a great presentation that you have made. Still on collaborations. I just came from Europe on Sunday. I was there for about 2 weeks, and there are a lot of opportunities between North-South and South-North. But one of the conditions is that the universities within a country, say like Kenya, first of all, also must have collaborations among themselves. The same to universities in Europe. Then the funds, as you are aware, are so many. So what are your thoughts? How are we going to move forward from here? I think you are happy. You are already working with, most likely with some of my people here, or you know they are here. But how do you want us to go? Do we do an MOU between MKU and MUST? Or do we start from your research lab and my research lab team here? And then what components, what kind of things do you want to see in that collaborations? I was thinking, like some of those webinars when you are doing them, I think we can work together on that.
Sure.
Can participate. When we are teaching, there's this new word that has become so popular in the last 2-3 years, what is called COIL, Collaborative Online Integrated Learning.
Yes.
You can give lectures to our students, especially who are in this, is this molecular or this diagnostics, and some mathematicians, if I make mistakes, understand.
Yes, yeah, you are right, you are right.
And then our people here can also teach with your people. Do you think we can look at collaboration in terms of supervisions of students? You know, those kind of things at Masters and PhD. Do you think we can work together in terms of sourcing for research grants jointly, those kind of things? Just your thoughts on that. But thank you for coming.
Thank you,
Very
Prof.
happy to see you.
Good. I think we can give him time to respond to those questions before we can get some more questions.
Yeah.
Go ahead, Prof.
Yeah, thank you very much. I think I'll take the questions in the reverse order. Yeah, so Prof, I really want to first of all appreciate even you coming to listen to this presentation. I mean, all of us are aware that in this country, getting the Vice Chancellor to come and listen to, a research, seminar, even something like this is not usually common. So I really want to commend you for that to start with. And just going to your question is, collaboration is really at the heart of research today. There's no way you can sit somewhere alone and do some, cutting edge, ground shaking work. I think that era is over. We have to act together, because even funders, the people who give us money to do the work, if you are applying alone and considering the complexity of the challenges we face today in the world, you need different brains. You need people from interdisciplinary, from other disciplines to come together and make the things work. So we are very collaborative. This is, I think we are, Frank can tell you about this. We have an open door policy ourselves. And it is just, if you look at the repertoire of collaborators that we have from Africa, be it Nigeria, Ghana, Uganda, Rwanda, South Africa, Egypt, Tunisia, that is just in Africa. And then from Africa, we have collaborators in all the continents, from Australia to South America, and not talking about America, North America, and even Europe. So number one, we have to be collaborative. Number two, especially for us in Kenya, we have to create time. We have to be available. We have, you see, I have to be willing to sacrifice the day and come all the way from Thika to Meru, okay, and make connections. So, because I realize this is one of the major, we may underestimate it, but it's a serious problem. I know some of my friends from the University of Nairobi, we can take months without meeting. And in fact, they usually have time for even a Zoom meeting when they're in America, not when they're in Nairobi. But we are just like 40 kilometers, away. So we have to put resources, as I'm so happy because the top university administrators are here. You as the administrators, as the Vice Chancellor, as the Director for Research, you really need to put money. You need to, if Frank says they want to go to Masinde Muliro, you need to put in some money for his fare, for his subsistence going there. And it should not be a pain for the researchers to make connections locally. Okay. I know sometimes when we are going to South Africa or Europe, it looks glorious, and the universities may even put money. But when we are going to local universities, people may think, oh, why should we invest? But we have to invest to get the results. And just to reply to the director directly, we are open. We will collaborate with you. Frank knows. I really want to thank Frank, because he's made he's taken the steps. He came to my office. We had discussions. We had some problem with Oxford Nanopore. He sent his staff, and they came and they taught my guys how to use the equipment. And now we have data that we are analyzing to publish together. So that is a classical example. Now, coming to Frank's questions, so you talked about the IFAST. So in the intermediary chambers, we have, this has oil, PBS, oil, PBS. Okay? So immiscible fluids. We know oil and water does not mix. Okay? So by creating these interfaces, you can be able to, as you pull your analyte of interest, it can be washed. Okay? So these are washing chambers, sort of. So, in the lab, classically, we wash by putting maybe PBS, we centrifuge, discard spanatant, wash again, these kind of things. So with this, you avoid that scenario. So, and by so doing, you can purify your analyte of interest. And about enzymes, is nice you mentioned that, but because as you speak, we have an initiative in our lab to start doing enzymes locally. So this is a discussion we can follow up, because we do a lot of LAMP work. We do a lot of PCR work, TAC polymerase. Is here when you are buying something locally from these suppliers around, the price is usually like 3 to 4 times what people in North America buy for. But you can understand, people put markups, taxation, these things are coming by flight. I mean, it's more expensive. So research is more expensive here. But if this, as Frank mentioned, these reagents are not complicated. It's not rocket science to manufacture these things. So with our new lab, which you should visit, is top of the notch. If you come and close your eyes and then open, you'll not know whether you're at University of Tokyo or Harvard or London School, because it's top quality. So we want to start making enzymes locally, and our aim is not to sell abroad. Our aim is to support researchers in the region, and if we are lucky enough, in the East African region as well. So thank you.
Thank you. Thank you very much, Prof. Now, listening to your presentation, I realize IFAST is like an amplifier of a cash-based test. Now, the question would be, without this amplifier, what would be the sensitivity of the cash-based tests? And with the FAST, how much do you improve it? Because that is key to determining whether it is cost-effective and whether it has any value in terms of it being used as a strong test for detecting this infection. The second question which I am asking is that now that you have done this and we still have the issue of COVID with us, how applicable can this test be used in our health facilities? Do we see it having such a robust application to the level that one, it is not very costly, second, it can be utilized under different field conditions? Because you said after the 7th day, it loses the value. Thank you. Please address this.
Thank you, Prof, for very nice questions. I think, so what we've done is, when you're talking about a molecular diagnostic, there are usually several big challenges. One of the biggest challenges is sample preparation. Okay, as you may know, like extracting DNA from a sample, you need kits. I mean, in the labs, we usually use these QIAGEN kits or Zymogen kits or Zymo. I mean, there are many companies that in Vitrogen that produce kits for DNA extraction. But you see, these kits are expensive, and they sometimes require centrifuges and other big equipment that a small lab may not be able to have. So the IFAST technology, the device itself, enables you to do DNA extraction on chip, and then you can amplify on chip. You can multiply your DNA sequence to many copies, okay, and then be able to visualize this. So this is key. So the innovation itself is the ability to bring a test into something that is field deployable. Okay, yeah. So you asked a very nice question about. As you can imagine, we were developing these tests from 2020, where thinking about the challenge that the pandemic was posing, okay, and looking at the diagnostic challenges that were there then. But as we know, the pandemic is literally on its tail end, isn't it? So we are now taking this test to look at other diseases. Our current focus is on cryptococcal meningitis, cryptococcal neoformans, which causes meningitis, and also maternal bacterial infections. I mentioned about GBS, group B streptococcus, but also we are looking at Neisseria gonorrhea, which we just published a few months ago, and other diseases Okay, so we want to, and even neglect tropical diseases. We want to now see, can we use this technology for other diseases of importance in our setting? So this is the goal. Lastly, your question is about ability to now take this to the clinic. Is a very loaded one, because this needs a sort of an ecosystem approach, because we are I can say, we are asset developers. So we think about the molecular biology, and then we combine with engineering to come up with the test. But we may not be well skilled to now take this test to the market, to the regulators, to find companies now to pay for, to maybe pay us some licensing amount of money, and then for the companies now to sell to the clinics in Karachuonyo and Meru and other places. Okay, so we need to think. This is why as scientists, we need to think about how do we stimulate a vibrant biotechnological industry and even have commercial people. Some of our students should not be in the lab. They should not stick to the lab. They should go out there and become business entrepreneurs. A student should come and tell me, oh, Jesse, can we take this test? So like for GPS, we've patented this, okay? And for the others, we are thinking of patenting. But now the student should take this, or any other person, and now go commercial with it. Okay, so as academicians, sorry to say, sometimes we are not skilled in being able to bridge that sort of valley of death. Okay, taking something from the lab to the clinic. We do very well with grant money. Okay, when the donors are giving us money, which is fairly, depends on how beautiful our proposals are. But we need somebody now to take this from here to the market, to convince KEMSA, to convince governments, to convince private hospitals to buy the tests for their own use. So this is really the elephant in the house. Is a big issue that we have to address somehow.
Probably the only bit that was not maybe handled is the sensitivity part of it. There is a cast that can detect COVID, but with the fast. I was wondering how much does it improve the diagnostic capability with or without the fast, fast?
Yeah.
And what is the level of false negatives?
Okay. Yeah. So because this is a lab-based test. I mean, we're just doing laboratory validation. Our sensitivity, we were looking at in terms of detection of the lowest number of genomes per mil. So as you can see here, we were able to detect 4.7 genome copies, okay, per mil. This is quite sensitive if you think about, comparing with, RDTs, and even with RT-PCR itself, this is very competitive, okay. So, but the work we just published on this, because this work now we already published. We compared with clinical samples, and the sensitivity was good, around 90%. Yeah.
Yes, Prof is sharing with us a very important data. Any other questions? Yes? Did you? Okay. Thank you. Let us appreciate Professor Kitaka for the good work which is done and for the good presentation that he has shared with us. Thank you very much, Prof, and we welcome you to Meru University to continue sharing with us your good research. And you've had the avenue is there now for further collaboration. Thank you very much, and karibu.
Yeah, thank you, Prof. I just wanted to go back to a question the Vice Chancellor asked on what step this takes. I think we can institute, we can already start doing a memorandum of understanding, and we can even do joint supervision of students. You talked about COIN. We are very happy to collaborate on that. Fortunately, I'm the director for grants and development, and if I'm able to push an MOU, it also boosts my own reputation within the university. so it's a good thing. So yeah, I think we can take the discussion. Yes, super nice. Thank you, Prof. Really appreciate it. Thank you very much.
Again, let us appreciate our professor for the good presentation. Okay, moving on, we have lined up other presentation. Yesterday, within the session of climate change, adaptation, mitigation, and resilience, there was a presentation which was not given. We've lined it today. And at this juncture, I'm asking Faith Goody to prepare herself so that she can come and share with us research work and make the presentation to the audience. I believe you've already given your presentation to our communication team. Thank you. Karibu. So this presentation is slightly different from what is already being shown in the schedule. We had to look for ways of how we can accommodate this presentation because it was supposed to be there yesterday, but for certain reason it couldn't happen. Thank you very much, Faith. We're very grateful, and we apologize for not having you give your talk yesterday. Please, the floor is yours. You can share with us your results.
Thank you.
Yes.
Thank you very much. My name is Dr. Faith Nguthi. I work at KALRO. I'm based at KALRO Thika, currently based at the National Sericulture Research Center. And I've worked in KALRO for many years. I'm on my tail end of retirement. And as Professor is saying, this is a complete detour from what Professor has presented, because here we are going down to the ground with working with the farmers. So my presentation is on work that we did, validating and disseminating climate-smart banana technologies using the Farmer Field School approach. And probably for this audience, it may be, I don't know whether it might be something new. How do you move down here? This. Oh, okay. So we did this work in 2 counties, in Bomet and Tharaka Nithi. And for my introduction, we are saying that Kenya's agriculture is central to meeting challenges of feeding a growing population, creating wealth, reducing poverty, managing degradation of natural resources, and also mitigating climate change impacts. Climate change, the impacts of climate change on agriculture. I think we have all seen these kind of pictures where the climate change is characterized by erratic rainfall, increased temperatures, increased frequencies, intensities of droughts and floods, and increased incidences of plant pests and diseases. And it is projected that climate change will cost Kenya 3% of GDP per year by 2030 and 5% by 2050. And rain-fed agriculture is one of the culprits of climate change, and it is adversely affected by these extreme weather events. The government of Kenya has taken initiative and is partnering with various development agencies to respond to these challenges. And one of the projects that hosted this work that I'm presenting was a Kenya Climate Smart Agriculture Project, KSAP, that was funded by the World Bank. So the objectives, the overall objective of the KALRO KSAP Banana Value Chain Project was to enhance banana productivity, build resilience to climate change, and contribute to carbon dioxide sequestration. The specific objective was to validate and disseminate climate-smart banana technologies, innovations, and management practices. Over the years, KALRO and other agricultural research institutes have been able to come up with the technologies that can mitigate climate change impacts on agriculture. But most of these technologies have remained on the shelf and are not getting down to the target clientele, who are our farmers. So through this project, we took the initiative to get this, what we are calling teams, down to the farmers. And we used a banana. And why did we use a banana? Banana is my pet crop. I've worked on banana for many years. I was involved in the introduction of the tissue culture banana with Professor Kahangi and Dr. Florence Wambugu. We have worked all over the country getting the banana, the tissue culture banana, to different parts of this country. So it's a popular food. It's a good source of carbohydrates, vitamins, and minerals. The surplus production gives reliable income, and thus ensuring household food security and welfare. And once planted, the crop can take up to 8 years, still giving the farmer a good crop if the proper management is taken. It can also be intercropped in the early stages with other crops, and it is valuable also for environmental conservation. But there are various constraints in banana production, and one of them, which is key, was the unavailability of clean planting materials of adaptable varieties, because what farmers were doing previously, and they are still doing it even today, is to exchange planting materials which they get, suckers, which they get from their farms, and take them to, and so the diseases that are already in the soil and the pests, they're just spreading them all over the country. And then we also have a problem of the low and declining soil fertility. Soils in Kenya, the farming soils, have been used over the years. We also have high incidences of pests and diseases. And with the emergence of climate change, we are getting many new pests that were not there. And so there is an increase of pests and diseases. We have also high post-harvest losses of the banana. And we also have limited knowledge. Farmers have very little knowledge on how to grow the bananas in the right way. And I think we have already mentioned the weather patterns and the poor marketing channels. So the climate smart banana teams, that is technologies, innovations, and management practices, that we worked with the farmers that we were working with in Bomet and Tharaka Nithi, included, the key one was clean planting materials, which we did mainly tissue culture. And for the sake of those who are also doing suckers, we showed them how to be able to clean those suckers so that they can reduce the diseases and pests. We also came in with improved banana varieties. We also looked at integrated soil and water management practices. We looked at integrated pest and disease management practices. And we looked at post-harvest handling practices. The methodology we used is a farmer field school approach. This is an approach that has been used in Kenya over the years. It was introduced in 1995 by the FAO, and at that time it was focusing mainly on IPM. Today, the farmer field school is widely used on many other technology dissemination activities, and the approach is based on learning by doing, and it is usually done along the full crop cycle. So as I was saying, it has expanded to other disciplines. We started with IPM, and now it has expanded to many other disciplines, including land and water management, beekeeping, fisheries, many other disciplines, including HIV and AIDS. And recently, the climate change has been included in the FFS learning curriculum. Just recently in 2021, now there is a curriculum that is handling climate change. So we did our work in Bomet and Tharaka-Nithi. We had 2 sub-counties in Bomet, Longisa, in Bomet East, in Longisa Ward, and in Sotik sub-county in Danai Ward. And we worked with common interest groups in those 2 sites, and we worked with a group, 2 groups, self-help groups, and those are the details. We also worked in Tharaka-Nithi, in the Igamba-Ng'ombe sub-county, and also in Mara sub-county. And we worked with the groups of farmers whose main interest was growing bananas. Because the KSAP project was working in different counties all over the country. So these were groups in those counties that had expressed interest in growing the banana. And these are the steps that we followed in the FFS implementation. We did site selection, and we also selected the common interest groups. And these common interest groups are those that had expressed interest in growing the banana. We did a baseline survey also to find out the situation on the ground about bananas and how farmers were growing bananas and their challenges, the varieties they were growing in those counties. Then we developed a curriculum, FFS curriculum. And then we trained facilitators on that curriculum. And the facilitators were mainly drawn from the extension offices and also some of the lead farmers in those areas. Then we went ahead and established the FFS. We established the FFS's farms with the farmers. We trained, and from there we started training the farmers from planting up to harvest and also value addition of the banana. So we worked with them the whole plant cycle. And the cycle for banana, depending on where it is being grown, is usually, like in Tharaka-Nithi, the bananas were ready by 8 to 9 months. In Bomet, the bananas were ready by, some were, like in the last one of the sites, they took almost 14 months. So we worked with those farmers along the value chain, training them and working with them. We also did, the agro ecosystem analysis is a key aspect of the of the farmer field school. This is where the farmers, we gave them, like we selected 10 plants on which they were going to collect data, because you don't want to burden the farmer with a lot of data collection. So you give them 10 plants on which they are going to collect data along the crop cycle. So they collected data, they recorded that data, and we helped them analyze that data to be able to know which varieties were doing better. And yeah, so I will come to that again. So we also did a market survey, and to find out, so they are growing these bananas, where are they going to be growing them? And we linked them to traders in their region. And then one of the other thing, which is very key for the FFS, is a graduation. It is a point where farmers come and they invite other farmers in the region, and they're the ones who are now training these other farmers who are coming and taking them through the training that they have gone through. So this is what I'm talking about, the IESA, the Agriculture Ecological System Analysis, where the farmers meet on selected days and regularly, and they observe the crop, and they make their data and their observations. Like you can see, one of the farmer there is measuring the height and the width of the flowers. And then they record the data. We showed them how to record the data. And these were some of the results from that IESA analysis. So farmers were able to go along with their crop. Here I've only presented the data on days to flowering, days to harvest, and bunch weight. They also took data on the number of leaves. They also took data on the width of the stem as the crop progressed in growth. So they were able to see which varieties mature faster. Among the three, we gave them 3 varieties. Which ones mature faster than the others? Which ones have a higher weight than the others? And, sorry. That one is not coming very nice. But I think what I'm talking about in this slide are the impacts of the results and the impacts of the work that we did. And I'm saying that farmers were able to identify the different banana varieties and their advantages, and they were therefore able to make informed choices on what varieties to grow. And what we are seeing currently, that the farmers are now using the TC planting materials in those 2 areas, and majority are aware of the technologies and innovations and management practices that we introduced to them. And we saw that what happened is that all the 4 FFSs that we were working with became model training farms for farmers in those 2 counties. And some members of the CIGs have also become TOTs and are invited to train other groups in other counties. And what we found is that farmers have now an alternative food crop. Farmers, especially in Bomet, were growing maize. And maize was seriously affected by the maize lethal necrotic disease in Bomet. And so they didn't have enough food. They didn't even have enough source of income. But when we introduced the banana, the TC banana, they are able to, they told us they eat that food, they sell, and they get money in their pockets. So and you know, the maize lethal necrotic disease came mainly because of climate change impacts. So we found that probably we were mitigating by introducing this other crop, the banana, which is not affected by this other disease, and the farmers are able to eat, to put money in their pockets. Yes. And here what I'm saying is that although the FFS was one crop cycle. Some of the CIGs that we worked with are continuing to work together and have formed producer organizations. For example, in Bomet, the farmers have formed a banana cooperative society, and by December 2020, they had over 300 members who had been registered. And actually, the secretary of one of the CIGs that we were working with is the secretary of the cooperative. So these are some of the results. I'll just browse over them. This is what the work that we did. We also did water management, drip irrigation. We trained the farmers on how to harvest. We trained the farmers on how to do post-harvest by providing them with the ripening chamber. We held field days for other farmers, and it is the members of the CIGs who were actually training these other visitors on how to handle the banana. And then we also had the graduation for the farmers in the 2 areas. And thank you for listening to me.
I don't know how to operate this. Thank you very much, Dr. Goody, for the very exciting and practical presentation that you've shared with us. And we need to appreciate Dr. Goody for the good work which she is doing. Let us give her a good clap. At this juncture, now I open the floor so that those who may be having comments or questions, they can direct them to Dr. Goody for the good presentation she has done. Okay, probably I can.
Thank you, Chair, and Daktari, the presenter. We come from different backgrounds here, and I must appreciate that I've learned what it is that exactly call technology in agriculture, some aspect of it though. And yeah, my background is in electronics, and so I have some concern as I appreciate your good presentation. I've followed, although I might have missed some parts, but my concern comes in here. When you mention technology and the challenges you have experienced through the study, data collection, among others, I think it would have been much helpful if through the scope of your audience and target, you thought of using the real technology that people in computer science and physics study in electronics. So the concern is, do you think of collaborations with people from different disciplines as you go or carry on with the project to reach the communities? And this is very helpful in my view. Give me some seconds, please. Because one thing that I'm concerned is, to what level do you intend to take the project? And how do you view the future of your project, considering that we need to talk about sustainability? You mentioned diseases are cropping due to climate change. Of course, yes. You'll find a disease that was not inherently in a place at any given time. How do you keep track of these diseases in real time to avoid lab work that takes much time? How do you keep track of nutrition? How do you keep track of other forms of stress with an ease way that can help you access the conditions of the crop the same way? So do you intend to integrate the traditional agriculture technology with this kind of technology I'm speaking about, the use of smart agricultural technologies, use of sensors, precision farming, and such kind of things? If you are to do so, please, it would be very interesting to think of cooperating with people from different subjects as you work out the project.
Thank you so much. My question is one. I come from Kisii, and usually when a banana starts yellowing, there's no remedy for it. So I'm interested to know if there's a remedy for a banana that has fallen sick, if I can use that term.
Thank you very much, Daktari, for staying until today. I know we had a lengthy talk with you. I really appreciate your patience and your cool. Mine is, I'm also in a different field, but involved in a study that includes bananas. So I don't know if there is any correlation, and you might need to educate me on this. Everybody is trying to do sanitation pads using bananas. It's way out of your line, but I don't know if there is any similarities. And within that study, they're taking various types of stocks to actually build the sanitary pads. Now looking at the toxicity and looking at that. And that is what we are doing now, collecting them from different parts. Is there any correlation or what you'd advise that these ones from this area would make good pads? Thank you.
I think we give our time to respond to those questions, then we'll come to you.
Thank you for the question. I think I'll start with the one from Prof. about the sanitation. I've heard about that, and I know that people are making sanitary pads from the banana fiber. That's the stem that produces very good fiber. And I think that's a great thing, because we are also trying to look, it is interesting to also look at value addition. That's part of value addition of the banana, because what happens is that farmers feed their the stem to their cows. So if there is a way that we can value add on the stem, and it is being done actually, I know it is being done. Is it in Uganda? And even Kenya, they are now starting to do that also, I think in Western Kenya. So it is value addition, and it will bring in money. I think what we are looking for is putting money in the pockets of the farmers, which will also make them food secure. So I think that's a legit study that you're doing. Let me get to the technology person up there. Well, we would be very interested in collaborating with the tech people to help us. What we are doing is, we are farmers and we are not in tech. We tried to bring in technology here in this work, at the very minimum level of developing a mobile app for the farmer. Actually, I didn't mention that, but we developed a mobile app which is hosted on Play Store, called Smart Banana. And that mobile app is very simple. We are just communicating we are giving the farmer an opportunity to connect with the scientists, to ask questions about whatever challenges they are going through on their farm. And they can be able to send pictures, they can be able to send questions, and the scientists are able to pick that and answer those questions. It has not moved very far, but that is the farthest that we went with a technology. And we would appreciate, we can have a talk later and see how we can incorporate your technology. Thank you. The other question was, what was the third question? Oh, yellowing, yes. There's somebody who talked about yellowing, and in Kisii. I've actually worked a lot in Kisii. We introduced the tissue culture banana in Kisii many years ago. And yellowing can be caused by many things in banana production. It could be nematodes. It can be caused by nematodes. It can be caused by weevils, which eat the roots. And so the plant is not able to take up nutrients, and so it starts yellowing. It could be a disease called fusarium wilt, which affects the leaves. It could be by a disease called sigatoka, which also affects the leaves. So depending on that, the farmer needs to know. We need to know what is causing that disease. And so some of them have cure, like the nematodes, you can control them. The banana weevil, you can control them. Even sigatoka, even the other disease, that fusarium wilt, is very difficult to control because it's a bacterial disease, and so it affects the roots and the crop dries. But there are varieties that are resistant to fusarium wilt. Yeah, so you would need to know why your bananas are turning yellow and dying. Thank you.
Yes.
Oh, okay.
Thank you, Professor. My name is Dr. Nabea. I come from Chuka University. I'm also heading the media team, which is transmitting this conference live. I come from the Rakanivi, and this research, Daktari, has actually interested me. It's quite exciting. But I'll be very excited if I would know the exact location of your research and the justification for it in the Rakanivi.
I can answer that. Yes. Maybe you were not there when I said where we were located. Where we carried out the work. We were in Tharaka-Nithi, in Igamba Ng'ombe sub-county, Mariani ward, and Mara sub-county. Mara down there, in those dry areas. And they were selected because we were looking at areas that have been affected by climate change. So these are dry areas. Is that okay? Is that true? Because you're from Tharaka-Nithi. Yeah, and then we worked with the county government. We worked with the Tharaka-Nithi county government, and they're actually the ones who directed us to those sites. And we actually worked with groups that they had already pre-selected who had expressed interest in banana, because there were many other crops that the project was handling. So these were groups that had expressed interest in growing the banana. Yes. Have I answered your question? Any other?
Thank you very much, Dr. Faith Gudi, for the good presentation. We're very grateful that you are able to wait until you shared with us these results. I think there could be some other questions, but if you have issues or comments, Faith is going to be with us. You can still forward some comments to her. At this juncture, thank you. Let us appreciate Dr. Faith again for the good work. At this juncture, we have lined up an online presentation. So we just move to that presentation so that we can listen to Pauline karaoke. We are going back to our schedule again because we had to slot in Dr. Gudi. So the third presentation is what we've lined up. And the technical team, you can put her on. We can listen to her. Morning. Can she hear me from here?
Yes, I can hear all of you.
Okay, thank you very much, Pauline. We welcome you to this conference. We're very eager to listen to your presentation, and the floor is yours as long as the technical team gives you a go-ahead. Thank you very much, Dr. Pauline Kariuki, and welcome to this conference. You can hear me, Dr. Kariuki? Hello? Is she?
I have a presentation I'm making today. Good morning.
Morning to you, Dr. Kariuki.
Are you able to get me?
Yes, we can hear you very well. We can hear you very well, Dr. Kariuki.
Do you have any other 5 rooms around here? Hey, which one?
Dr. Kariuki, you are very audible to us.
Hey, light.
Dr. Kariuki.
Hello.
Hello.
Yes. Hello. Sorry for that interruption. I'm now ready to present. Good morning. My name is Pauline. Are you able to get me?
Yes, we are getting you. Just continue.
Yeah, my name is Pauline Kariuki, postgraduate student at Technical University of Kenya. I am humbled to make my presentation to your conference, the second annual International Research Conference. My topic is on the contribution of research and innovation and development of Kenya. The background information is the COVID-19 pandemic has brought to the forefront the crucial role of research and innovation in the development of health technologies to address global pandemics. The development of diagnostic tools, vaccines, and therapies have been critical in managing the current pandemic. According to a report by the World Health Organization, innovation and research have played a significant role in developing new technologies to address the COVID-19 pandemic. That is according to WHO 2021. So this paper aimed at exploring the contribution of research and innovation in the advancement and development of healthy technologies. Then motivation. What was my motivation, or the statement of the problem behind my research was that despite the significant contribution of research and innovation in the development of health technologies for addressing global pandemics, several challenges remain. One of the challenges is the lack of adequate funding for research and development of health technologies. So according to the report by the World Health Organization, the lack of funding is a significant impediment to the development of new technologies for addressing global health challenges. That is WHO 2021. The limited funding has also affected the equitable, has also affected the has also affected the equitable way of coming up with research and development innovations. Another challenge is the slow adoption of new technologies in low-income countries. Although research and innovation have led to the development of new healthy technologies, the slow adoption of these technologies in low-income countries has hindered their impact. According to a report by the World Health Organization, the lack of appropriate infrastructure and human resource in low-income countries has hindered the adoption of new technologies, that is WHO 2021, and this has resulted in a significant health gap between high-income and low-income countries. So that was my motivation. And then research objective, the general objective was to explore the contribution of research and innovation in advancing health technologies to combat pandemics. Now, specific objectives of the study were, number one, to examine the role of research and innovation in developing health technologies for addressing global pandemics. The other research objective number two was to identify the opportunities of using research and innovation to develop health technologies for global pandemics. Number three was to identify the key health technologies that can be seen as a result of that have been as a result of research and innovation efforts. Then finally was to analyze the impact of health technologies in managing the COVID-19 pandemic and provide recommendations for policymakers and stakeholders on how to factor in research and innovation in health sectors. Then the research methodology, the research was done in selected counties in Kenya, which included Nyandarua, Kiambu County, Kitale, and Kisumu, which were purposefully sampled among the counties in Kenya between January 2023 and March. The research focused on exploring the contribution of research and innovation in advancing health technologies to combat pandemic in Kenya. The researcher used a structured sampling technique of health professionals who added up to 1,200 respondents. A quantitative research design was employed. Open access questionnaires were used to collect data, and the data was analyzed using the SPSS software. So what were the major outcome or findings? One of the findings on the respondents was that on the role of research and innovation in developing healthy technologies for addressing global pandemics, 65% strongly agreed that we can have our research and innovations being incorporated in providing technologies that would address the global pandemics. 25 agreed, 5 were neutral, and 4 were disagreed. Then objective opportunities of using research innovation to develop healthy technologies for global pandemics. Majority, that is 40% strongly agreed, 56 agreed, and 3 were neutral, and 1 was disagreed. And then lastly, we had research and innovation have regulatory frameworks for developing healthy technologies. This was a major concern, that we do not have regulatory frameworks provided by the government or our stakeholders in the Ministry of Health for developing healthy technologies to curb the global pandemics, and therefore 10 of them strongly disagreed that we do not have these regulatory frameworks. 1 disagreed, 10 strongly agreed, 1 agreed, 4 were neutral, 70 disagreed. So majority disagreed, and 15 strongly disagreed that we do not have the regulatory frameworks for developing healthy technologies in our research and innovations. Let me move on and look at the conclusion. So the study concluded that research, sorry for that. The study concluded that research and innovation is critical for the development of effective health technologies to prevent, detect, and treat pandemics. The majority of respondents agreed that research and innovation is essential in the development of healthy technologies for addressing global pandemics. However, the study also found that the development of innovative healthy technologies to address global pandemics remains a significant challenge in Kenya. Recommendation. One of the recommendations of this study is to promote collaboration between research and innovation stakeholders and the health sector specialists in developing healthy technologies. Number 2 recommendation is to establish the establishment of regulatory frameworks for the development of healthy technologies by the governmental agencies and non-governmental organizations. Number 3 is to promote research and innovation in addressing global health challenges, including pandemics. Thank you for listening. Thank you very much. Thank you.
Thank you for that good presentation, Dr. Kariuki. You've given us the way we view the contribution of research and innovation, and I want to ask my audience to appreciate the good presentation that you have given us. Please let us clap for Dr. Kariuki for the good work. With that, I want to open the floor so that if there are any comments or questions, then they can be directed to Dr. Kariuki. Any questions? Yes, Dr. Kariuki, we've got somebody who is interested knowing this. But before they can ask, I've got 2 questions probably that I can direct to you. You've said 65% of the people strongly agreed that the technology inclusion in health technology is important. But you also indicated that 25% strongly disagreed that technology should not be included. The issue is who was your study subject, because the level of knowledge is very important here. And secondly, did you get any reason as to why people disagreed, strongly disagreed that technology should not be included in new health innovations? Thank you.
Thank you very much, Prof. I want to submit that the first one is that research and innovation in developing health technologies for addressing global pandemics, they agreed that we need the research. That was the approach, that we need research. The healthy professionals, the healthy practitioners agree that we need the research. 65% agreed we need research and innovation. But now when we come back to looking at, do we have the regulatory frameworks to coming up with these health technologies, they say that we do not have the regulatories. But now 65 agreed they need research in healthy sectors, they need innovations. We need to come up with research, more research oriented healthy matters and innovations that can curb these pandemics. 65 agreed and 25 agreed. Only 5 were neutral and 4 were disagreed. But now coming back to asking them whether there are those regulatory frameworks that can help us cooperate or collaborate between research institutes and innovations with healthy sectors, they found that there were no regulatory frameworks. So what we need is the regulatory frameworks to inform us how do we go about coming up with research and innovations in the health sectors. Professor, I hope I have answered. I have tried.
Yes, you've answered my question, but I think it is an issue of us knowing where we can get that information, because I know that there are certain regulatory bodies that have been formed to regulate the emergence of various technologies that concern health. For instance, we've got NACOSTI, we've got intellectual property right policies, and I believe if we were to create more awareness, then those the persons who are participating in your study would be aware of where to source for such requirements in future. And I hope it is part of your recommendation that creation of awareness of where to source for such regulations to the study participants is important. Any other questions? Good. Again, let us appreciate Dr. Pauline Kariuki for the good presentation that she's given us. Kariuki, we're very grateful. We're happy that you are able to give this presentation online. And we hope more comments or questions will be directed to you by people who may be having issues which are not clear up to now. At this juncture, I think we've already done 4 presentations.
Thank you very much, Prof. I also want to say thank you for having me online and enabling the ICT people, enabling us to have a very smooth presentation.
Yes.
I am so grateful. And thank you, Meru. Thank you, Meru University.
Thank you and welcome again to our future conferences or any other forum that we share research work.
Thank you. Thank you and have a blessed day.
Thank you. Thank you. So as I was saying, at this juncture, we've already listened to our guest speaker. We've listened to 3 of our presentation. And I think it is good for us, we take a break. After the break, we still have more work lined up for us, more titles lined for us. So I would suggest we take a break. Now it is what time? It is 10:55 according to my watch. We take a break of 30 minutes and we should be back here by 11:25, so that we can take on the other section of the papers that are lined up for presentation today. Okay? Okay, my co-chair is telling me it would be very good for us to confirm those who have already, who are here for the presentation after the health break. Anybody? I know I am aware of a few of our colleagues. Jarvis Meriti is there. Millicent, is Millicent? Yes, Millicent is already here. We have Zakayo Mwangi. Zakayo Mwangi is our colleague. Okay, I think we will contact Frank to get to know who is going to give that presentation. Do we have Peter Ntoiti Kailema? Oh, good. He is there. And then Dixon Mwenda Kenyua. Is Dixon Mwenda Kenyua with us? Okay, we'll find whether he's going to be available. Zachary Murioki is already here, so we have that Daktari on the wings, and Cindy Amogo is our colleague and should be here to give the presentation. So we have about 6 presentation, and we'll do it before we break for the lunch. Okay, thank you very much for listening, for coming to listen to the presentation. We look forward to seeing you after the health break. Thank you.
Oh, yeah.
Hello. We are serving tea at the cafeteria. Then for